on. So remember we talked about the secondary oocyte with its first polar body, and we had the zona pellucida around that. And this is what is released from the egg. And when the sperm fertilizes it, it passes through those follicular or granulosa cells. So I'm not going to bother drawing that because we're focusing here on what happens to the nuclear material. And it inserts into the egg. It inserts into the egg its nucleus, all right, and a couple of centrioles. And so over here we had the female pronucleus. That's, well, let me just back up a step because we don't really call that the female pronucleus until our second meiotic division has occurred. So let's back this up. So we have our 2N, 2 copies. And so as soon as this occurs, we see a second division, our second meiotic division, but we need to include our sperm head in that. And we see our second polar body, all right? So first polar body, which remember was also 2 parentheses in. Second polar body, which is now just in. Our sperm is also in, right? And now this is in because it split when we had the second meiotic division. So we have a female pronucleus, which is in, and the male pronucleus, which is in. So I had put links to videos up on my PowerPoints. For some reason with Microsoft, whatever current software is out, you can no longer embed videos. On my laptop, it doesn't even give me the opportunity to upload from the web. My office computer, it has the button there to click, but then it always says this doesn't work. So my flash drive is not recognized by my computer in my office. So I found the links and then I had to email them to myself, and that's what I was doing at the beginning of class here, was opening up all the YouTubes and opening up the links. And I have a video of, you can just barely see the pronucleus. Do you mind hitting the light right there under the pencil sharpener? This goes for a minute, but to visualize the pronucleus, we'll just play the first few seconds over and over a few times. So this is looking at a petri dish. This is the egg with the follicular cells. And then now you see these two. You catch those? I'll go back and show you again. So that was the moment of fertilization, the formation of the male and female pronuclei and their association with each other. So let me stop it here in appropriate times. So look at this region right here, all right? And over here, you'll see the 2 pronuclei. Fertilization is going to occur right about here. You won't actually see it. The sperm had injected, but you'll notice some disturbances in the cytoplasm right about there. Now here's this one, and there's the male one. This is a speeded up film, obviously. Now also we've got the 2 cells, so this normally takes about an hour. You can see that the zona pellucida right here is thickened. And we're going through all the various cells. So this is what's happening in the formation now of the morula, all right? So when we've got more than 100 cells that are very small in size, you can see that the zona pellucida is still in place, and we're forming mold. This is while the zygote morula blastula stage is moving through the oviduct, instead of in the petri dish. Now, see what just happened? So a hollow ball was formed. So that's the blastocyst or blastula. And so now we have an even more distinct separation of cells as they're becoming differentiated. Because remember, all these cells have what chromosomal pattern?
2N.
2N, okay? Some are single layer. They're exposed to the fluid inside the blastula as well as the fluid in the reproductive oviduct and uterine cavity. Over here we have a cluster of cells. This is exposed to the fluid inside the blastula but not outside. These are exposed to the fluid outside but not inside. And we have the ones in the middle that aren't exposed to either. So we're starting to differentiate where the cells are located and what the chemicals they're associated with. And we still have the zona pellucida around it, all right? And this is what ends up implanting into the uterine cavity. So if we go back to our PowerPoint image here. So that was the different components of cell division that you saw. Remember, all the polar bodies are still captured within this zona pellucida. And then we have the solid stage, and then just as it enters the uterine cavity, we form the blastula. Okay? Now this is on the average. Obviously, if fertilization, as I said yesterday, occurs earlier, this blastula stage may appear more up in the isthmus than in the uterine cavity. So now, as it's moving down through the uterine wall, and there's some evidence that the uterus can move the blastula around so it implants in the proper location if it starts to settle in where it's not supposed to. Obviously, that doesn't always work. Usually on the posterior wall, in this upper portion right here, all the diagrams will show it on the side, but it's typically in the posterior. It now is going to hatch. So the zona pellucida opens up, and we see hatching of the blastula, and it settles on the uterine lining with this inner cell mass layer bottom down. It kind of makes it top heavy, or bottom heavy, if you will. So it settles on that surface. It's really difficult to find, at least it has been for me, to find accurate, if you will, as well as clear components of implantation. So this is in Spanish. So I'm not sure if the sound is hooked up here. It's a 9-minute video throughout. I'm not going to show the entire thing, and I fast forward through part of it. If you understand Spanish, I'll just let it play. So that you're looking at the surface of the zona pellucida here, and now they're kind of showing artistic license what's happening to the cells inside. Some of the music has waltzes and romantic stuff going on, so you want to listen to these videos. So now it's so we're showing the inside of the morula now changing to the blastula. Since Spanish is a Latin language, the words pop up, you'll probably recognize some of them. So I did show the hatching. I have another one that I don't have time to show all of them, but I put links to them up online. So I'm going to fast forward a little bit because it does a really good, this may make more sense to you, the rest of the film after we've talked about implantation. So now it's settled on the surface, and it gets underneath the lining, so it buries itself, it doesn't eat the cells, it separates them away. So you can see that there's a change in tissue here that's growing out from the fetal tissue. That's what we're going to describe today. As it grows out, notice how the surface bulges out. This will bulge to the opposite side of the uterine cavity. So as the embryo is developing, it's staying in this wall and just bulges it to the other side. So we end up collapsing that uterine cavity. All right, you'll see that in diagrams. I'm going to explain these terms yet. Just I want you to see the image because I'm going to be drawing still pictures of what's going on. So we're going to be focusing on the yellow tissue around and what the changes are occurring in that when we talk about implantation, and then we'll come back today and a little bit on Tuesday and talk about this embryo development here. But you'll notice the uterine arteries. This is in the stratum functionalis, and that thin layer, the single layer of cells, develop out here to form multiple branches. They look like tiny little fine twigs that allow access of the fetal tissue to maternal blood. So they open up the maternal blood vessels. And the fetal tissue becomes directly bathed by maternal blood for the gas exchange. These are going to be called the chorionic villi. So they grow out, invade the blood vessels, actually send up fetal tissue. By fetal we mean derived from the fetal, not the fetus itself, but the placental tissue. And so they're forming, this is the placenta, so there's the lowest, the deepest region forms the structure that's actually the placenta. You can see it has longer columns that are growing down. These up here become flatter, and as they push the uterine wall out, they don't develop at all. So we have the chorionic villi, and we have the smooth chorion, or chorionic levy, which means smooth. So that becomes flattened. The lacuna there are the pools of maternal blood. Go to embryonic development again, and let's get to So this is from the outside of the placenta, and you're looking at those chorionic villi, which completely, so here they are, surrounded by maternal blood. To get increased surface area, they're going to branch. It becomes smaller and smaller and smaller. So in the mature placenta, we have these very fine, they look like, when you look at them in a dissecting scope, just like you took little roots of a tree or a plant and washed the dirt off, and that's what you see. And so you have this exchange of nutrients from the mother's blood to the fetal blood. Okay? So that's kind of the process that we're going to talk about first, just dealing with placental development. So again, I'll link this to put an announcement up on D2L. So let's go back and look at these diagrams, and if you don't mind hitting the light again, thanks. Yes.
Chromosomes from both the mother and the father?
Yes, so the placenta is foreign to the mother, and some women have difficulty getting pregnant because half of the chromosomes are from somebody foreign to her. There are proteins on the surface of the cells that she recognizes as foreign. So in most women, there is a suppression of the immune response in the stratum functionalis, part of what's called the decidual response. And there's fewer plasma cells and fewer white blood cells and other lymphocytes. That suppression of the immune response can be systemic, and so it's a time when a woman is more susceptible to developing cancers because her overall body's immune system in rejection, especially the T cells that are sensitive to cancer cells, are at a lower response, but especially in the uterine tissue. However, for some women that doesn't work, and they develop antibodies against those proteins on the fetal surface that came from the father of the child. And any implanted tissue from that father is going to activate an immune response as if she was vaccinated against it and result in rejection of the implanted fetus. All right, so here we see the blastocyst from the outside, and here it's been cut open so you can see it from the inside. On the left, what we have is the stratum functionalis. This is the uterine cavity, and these are the simple columnar cells that we talked about forming the uterine glands. Remember I said they were rich in glycogen because at first the embedding blastocyst doesn't have access to the maternal tissue. There could be some spotting here as capillaries are torn, but typically this is not a digestive activity. It's not eating or breaking down maternal tissue. It's kind of just spreading it apart, kind of oozing in between the cells and the connective tissue. All right? So we're going to come back and pay attention to the inner cell mass, but right now I want to illustrate the blastocyst and the placental structure. So we see a single layer of cells out here, and these cells, actually all the way through, and these cells are identified as trophoblast cells. After implantation occurs, we will differentiate between syncytial trophoblasts and cytotrophoblast, right? But at this point, they're all single cells, and each of them is a diploid cell. We're not going to see haploid cells until the gonads occur, and the female, we don't ever see haploids in the male gonad. Because remember, the male baby is born with spermatogonia, and those are going to undergo mitosis. But in the female, primary oocytes, well actually they're still diploid, but they're in that 2(2) stage. So we don't see the haploids until puberty occurs. On the inside of this, we have those separate distinctive group of cells. I'm trying to deal with this limited board that I have, known as the inner cell mass. Okay? We're going to talk about a couple of layers of that. So this is what settles on the uterine surface, uterine glands, and so on. And I'm going to, if you have these images, I'm going to keep erasing and redrawing, so I don't know if you want to just take pictures. But it'll take too much time if I have to redraw everything. But this is the stratum functionalis. So although the blastocyst is formed and appears within the zona pellucida, by this time hatching has occurred. The zona pellucida is gone and the polar bodies are gone. One of the videos I'll put a link to, we don't have time to watch all of them, but it illustrates, it's an animation, but it illustrates that hatching. All right, you'll see the blastula kind of oozing out of that cap. Now, these cells, just under the inner cell mass, yes?
What's the blue cells again?
The blue cells, sorry. The blue cells are the inner cell mass, sometimes referred to as ICM. These are going to form the amnion and the embryo. The trophoblast tissue that is on the surface of the uterine wall is what's going to become our placenta. Okay? All the other tissues don't develop as well. So it's going to be the one that forms the long chorionic villi that actually have the gas exchange and what we think of as the placenta when we see tissue that's delivered. So in this contact between the epithelial cells of the uterus and the fetal cells of the trophoblast, these trophoblast cells start to multiply. Okay? So we're in this layer right here, and they start to fuse together. Those cells, the trophoblast cells that have fused together, are now called a syncytium. If you want pictures, I want to make my first erasure. So the nuclei and the cytoplasm remains, but the cells have joined the plasma membranes, fuse and disappear in part. And so we have an appearance. We'll have a layer of cytotrophoblast on the surface here, but then we have this syncytium. So now we call the individual cells, whether they are here, or over here, when they're individual cells, cyto means cell. So those are the individual cells, and the fused cells are identified as syncytial. That means together. Okay, cells that are together, syncytial trophoblast. And that's what you're seeing here. And they send down almost like an amoeboid-like movement, all right, a flowing type of movement. They send column-like structures down into and separating the connective tissue of the stratum functionalis. As that occurs, the whole implanting embryo sinks down into the stratum functionalis, as well as the stratum functionalis kind of growing up around it. Now, rather than redrawing all of this, I'm going to move the uterine tissue up over this. So take another picture if you want. Yeah.
So as soon as it fuses is when they become this
Yeah.
cytotrophoblast?
Syncytial trophoblast means fused. Syncytium is a fused structure. Syn means with. Okay? And so it's just kind of like, well, unless you're a plant biologist, certain funguses grow as a syncytium. One large, something like the largest organism there, it's just one large thing that kind of flows underground and amongst the roots of the trees. And so these individual cells that were the trophoblast cells in this position join together, their membrane disappears, it's like two bubbles joining together and forming one large bubble. So all the cellular content is there, but this is really all one large cell, if you will. So now it's going to sink down in as it kind of separates those maternal tissues. And we're going to illustrate that by moving the maternal tissue instead. So here's our uterine glands. And they now form a solid layer over the top of the developing embryo and fetal tissues. The syncytial trophoblasts continue to form as the cytotrophoblastic cells multiply. They are added to the syncytium, and so it just gets more and more developed. And so more syncytial trophoblasts form around the embryo, but again, they're always most developed deep to this tissue, deep to the inner cell mass. So these are going to become more extensive. But then we're also going to see syncytial forming under these cytotrophoblasts here, now that it's completely deep to the uterine lining. There's just not as much room for them to grow because they're limited, all right, by that tissue. Here's the fibroblasts. round up the fibroblasts of the stratum functionalis, round up, they no longer appear like fibroblasts, and this is known as a decidual reaction in response to this embedding. As the columns are embedding, they come in contact with maternal blood vessels, those spiral arteries, the small arterioles and arteries, and as you saw a little bit in the picture, they invade them. So if we had a maternal spiral artery coming up here, let's make that a little bit wider. The syncytial trophoblast tissue opens it up and kind of flows along inside it. Okay, so better three-dimensional picture here. And so the maternal blood is now in direct contact with the fetal tissue that is the syncytial trophoblast. And so what I've drawn here in a longitudinal section appears as little pockets or pools of blood from other blood vessels and these are identified as lacunae. They're not lacunae in the sense of the chondrocytes and osteocytes that we've talked about, but these are pools. So now the fetal tissue is kind of floating on a lake of maternal blood. So the cells in here, at first it's just the fused syncytial trophoblast. All right, but we're going to see some further development as these become more mature. All right, if you want to take a picture of that.
The syncytial trophoblast, isn't that the trophoblast fusing with the cells of the stratum functionalis?
No, the syncytial trophoblasts are cytotrophoblasts that have fused together to form one large multinucleate, but only a single set of plasma membranes. So if I took 5 cytotrophoblasts and they all became one large cell, this entire thing is one large fused mass of cytoplasm. All right? So where we see the more complicated columns, these structures are going to develop further blood vessels, fetal tissue, because right now we're just kind of growing out, but we're going to be seeing fetal connective tissue forming in here. These are known as, chorion is a fetal tissue, formed here. This is going to be the syncytium plus fetal connective tissue. And as these branch, these are known as chorionic villi, or the chorionic villi. This area out here, notice, can't branch very much. It's under the pressure of the surface of the uterine lining. And so this is identified still fetal tissue as the chorion levi. And that can also be spelled like that. It means smooth. Okay, I'll write it up here for those of you who are in the back. So chorion levi, or so smooth chorion versus the branching chorion. So now the cytotrophoblast continues to multiply and add to the syncytial trophoblast, but it also starts to send down cytotrophoblast cells into those columns of syncytial trophoblast. And these cells now differentiate into connective tissue outside the embryo. So it's fibroblasts making collagen fibers, and fetal blood vessels, capillaries, are going to move down into these villi as well. So we have a column of cytotrophoblast. Which one shall I choose? I'll do this one just because it's up high for everybody to see.